Needed a wash. Must have had worse things on it.
Exactly. Good morning, everyone. What I'm going to talk about is, so we've heard about what it's like working in a hospital pharmacy. We've heard about how we can use chemistry to understand how long drugs will last, and we can use the chemistry to actually learn a lot about the fundamental properties of drugs. What I'm going to talk about is some of the work that's done in my lab trying to discover new drugs, trying to develop new ways to treat diseases where we don't actually have a treatment at the moment. The way in which my lab goes about doing this is by using genetics, by understanding our DNA, to understand different diseases where we may not have good drugs. The disease that I spend most of my time working on is Parkinson's disease. So hands up, who's heard of Parkinson's disease? That's pretty much all of you. Do any of you know somebody who has Parkinson's disease? There's a couple of you putting your hands up. So Parkinson's disease, just to give you a bit of background on what Parkinson's disease is. Parkinson's disease is a disease of old age, so it mainly affects people who are over the age of 60, and the older you get, the more likely it is you develop it. It's a neurodegenerative disease, which essentially means that brain cells or neurons in your brain start dying off. They degenerate over time, and more and more of them die the older you get. And in the case of Parkinson's disease, what that does is that it affects your movement. It slows down the rate at which you move. It causes you to shake, to develop what a neurologist, clinician would say is a tremor. It affects a range of different movement aspects. There are a number of people, quite famous people, who have developed Parkinson's disease. This is Muhammad Ali, who's the boxer. If you've seen video footage of Muhammad Ali, you'll notice that when he's moving, it looks as though it's almost in slow motion, that someone slowed down a film. And that's because of the cells, the brain cells that have died in his brain. This is Michael J. Fox, the actor. He's had Parkinson's disease for almost 30 years now, and the same kind of things are happening in his brain. In his case, the main symptom that he shows is shaking. If you see him on TV when he is not taking his medication, he has actually quite a violent shaking, quite a violent tremor. And finally, this is Billy Connolly, who got diagnosed with Parkinson's disease a couple of years ago. He's a comedian. And actually, if you're interested in learning more about Parkinson's, Billy Connolly has actually done a couple of documentaries talking about what it means to have a disease which slowly removes your ability to move. And one of the saddest things about Parkinson's is that at the moment, although we have drugs that can treat the symptoms, some of the shaking, some of that slowed movement, we don't have anything that can stop the disease in its tracks. So eventually, more and more brain cells will die to the point where actually you're not able to move at all. You're completely incapacitated, you become bedbound, and eventually you develop infections that shorten your lifespan really quite significantly. So once you have a diagnosis of Parkinson's, normally you live for between 10 and 15 years before you die of associated problems. So there's quite a serious problem here. Actually, we know quite a lot about what happens in the brains of people with Parkinson's. This is a cartoon of a human brain. What you're doing is you're sort of looking up into the brain, into a part of the brain called the midbrain. And this is very important for movement. So the brain cells in the midbrain are part of the networks that allow you to initiate movements. If you're sitting down, if you think about standing up, a chemical reaction occurs in some of the brain cells sitting in this area of the brain that stimulate other cells on the outside of the brain and that eventually connect to your muscles and allow you to get up and stand around and walk and so on and so forth. What happens in Parkinson's is that there's a very important part of this bit of the brain called the substantia nigra, and that's just shown here. What happens in Parkinson's is that very specifically those cells in that region of the brain start dying off. And actually, they die at a quite scary rate. By the time you develop a symptom, like the shaking, the tremor, or the slowed movement, actually almost 80% of the brain cells in that region of the brain have died. And what's shown here is what the substantia nigra looks like in the brain of somebody who has died of Parkinson's. Almost all of the cells in this dark-coloured region, the substantia nigra, are dead. They've gone. And that's what causes them to develop those symptoms. We know that those cells make a chemical called dopamine, which allows brain cells to talk to one another. It's a neurotransmitter. And when those cells die, they can no longer communicate with other cells, which causes the symptoms. So we know a lot about the chemical basis of Parkinson's. What we don't know about is why those cells die in the first place. And this is where the research that my lab here at the University of Reading comes in. And the way in which we try and understand this is by using family trees. So what I'm showing you here is a family tree. I'm sure you've all got a broad idea of what your family tree looks like, your parents, grandparents, and so on and so forth. So what I'm showing here is this is the most recent generation. These are the youngest people in the family. These are the parents, grandparents, and great-grandparents. And in this diagram, basically all of the circles represent people who are female, and all of the squares represent people who are male. If a circle has got a line through it, that means that individual has died. And actually, this may be a family tree that you recognise. It's actually the royal family tree. And what it's useful to think about when you're thinking about your family and about your relationship with different people in your family is you can use family trees to think about particular traits, particular characteristics that are shared between you and other people in your family. And I quite like the royal family tree because it's a very good example of this. If you go right up at the top here with Prince Philip, the Duke of Edinburgh, you've got Prince Charles here and you've got Prince William down here. And what you'll notice, in fact, increasingly so with Prince William, I was seeing some footage of him over in India on the TV last night. All three of these members of the royal family suffer from male pattern baldness. They're all suffering from baldness, and obviously this is something which is quite close to my heart. And we know, actually, that male pattern baldness is inherited. It's passed down from generation to generation. We don't understand all of how it's passed down from generation to generation, but there's a clear inherited part to that. And basically, information has been passed down from Philip to Charles to William that have caused the follicles on their head to stop producing hair and to die off. What this boils down to, and hopefully this is a concept that you're all familiar with from your studies, is the DNA in all of their cells, which passes down this information. So a lot of the characteristics that you see in members of your family and in people around and about are down to the fact that their characteristics are being defined by what's in their genes. So if you look at your eye colour, your hair colour, the blood type that you've got, these are all defined by the actions of genes encoded in your genome, in your DNA. So that's an example, a fairly simple example from the royal family. But we can use exactly the same kind of approach to understand human disease. So what I'm showing here is another family tree. This has actually been anonymized, so you don't know who's male and who's female. So each of the diamonds represents a member of the family. Again, this is the youngest members of the family here, the parents, grandparents, and great-grandparents. And the difference with this family tree is that all of the diamonds that have been filled in and are coloured black, all of those members of the family develop Parkinson's disease. This is actually quite rare. Most people who develop Parkinson's don't have any relatives who develop Parkinson's. But in this family, and this is a family from Lincolnshire, so just around the area of the Wash over here at the top of East Anglia, in this family, almost half of the people in the family develop Parkinson's. So this is quite rare. They were seen by a neurologist, and that neurologist actually worked with geneticists who, when looking at this family and seeing the fact that so many people in the family develop Parkinson's, suggested immediately that there was a genetic cause for their disease, and that there was something in their DNA that was being passed down from generation to generation that was causing them to develop Parkinson's. So colleagues of mine working over at University College in the centre of London extracted DNA from all of the members of this family that were involved in the study and looked at that DNA to try and identify a change in the DNA or a mutation that causes their Parkinson's. What they found, and this is a readout, a Sanger sequence trace of the DNA from these families, showing one change out of all the 3 billion base pairs of DNA in their genomes. There was one change that was found in all of the people who developed Parkinson's, but in none of the people in the family who didn't have Parkinson's. And that, after a lot of work and working with other families who have Parkinson's with a similar kind of characteristics, a similar kind of clinical symptoms, it was discovered that actually this is quite a common mutation. It causes somewhere around 5,000 cases of Parkinson's disease in Britain, There are about 125,000 people with Parkinson's disease, living with Parkinson's disease at the moment in Britain. About 5,000 of those have Parkinson's disease because of a change in their DNA and because of mutation in their DNA. What I've been working on for the past 10 years now is trying to understand what that mutation does. And it's a mutation in a gene called leucine-rich repeat kinase 2. It sits on chromosome 12 in the human genome. And we've been studying it to try and understand how one small change in the DNA for that gene can cause the brain cells in these people to die off and cause them to develop Parkinson's. One of the more interesting things about this gene is that actually it's got quite a famous person associated with it. This is Sergey Brin over here on the right. He's one of the co-founders of Google. About 10 years ago, his mum developed Parkinson's disease. And it so happens that his family is of Ashkenazi Jewish extraction, so a European Jewish ethnic background. And for reasons that we don't fully understand, there's a slightly higher rate of having this particular mutation in this particular gene in Ashkenazi Jewish people with Ashkenazi Jewish background compared to the more general population. His wife, or ex-wife now, is a geneticist, and she said, you might want to have your genes looked at. So he got his DNA sequenced, and they looked at the leucine-rich repeat kinase 2 gene, and they found one of those mutations that we'd previously identified in the family from Lincolnshire. Now, the difference with Sergey Brin is that he's in his mid-40s at the moment, so he is below the age at which you'd expect people to develop symptoms of Parkinson's. But we know with a fair degree of certainty that as he gets older, he is very, very likely to develop Parkinson's disease, probably about an 80% chance. For most people, the risk of developing Parkinson's over your lifetime is about between 1 and 3%. So he has a 1 in 5 chance of not developing Parkinson's during his lifetime. Of course, he's a co-founder of Google. He's one of the wealthiest men in America. And what that means is that he can actually do something about that. And some of the research in my laboratory is funded indirectly by Google through Sergey Brin, who donated money to the Michael J. Fox Foundation, and who fund research in my lab to try and understand what those mutations do. So every time you search on Google, indirectly and at a very, 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 very small rate, you're funding research in my lab. So feel good about that whenever you use Google. One of the other interesting things about this is that his wife, as I said, is a geneticist. She founded a company called 23andMe, and this actually comes back to pharmacies and what pharmacists are now doing. Because actually, you can go into a pharmacy in the UK, if you go into Superdrug, you can buy a kit from 23andMe, it costs £125, and you can have your genome looked at. They use a technique called DNA microarray analysis, which allows them to look at about 1 million different changes or polymorphisms in your genome to see whether you have one variant or another. Now that can be used for fairly mundane things like showing the DNA change which correlates with your eye colour. It can show you where your family is from, so based upon your DNA we can get an idea as to which country, which region of the world you're from. But you can also get information relating to disease. And one of the changes in the DNA that's part of this kit is actually the mutation that Sergey Brin has. So you can look and find out about your risk of developing diseases. Now, whether this should be available over the counter without any kind of consultation is another question altogether. But this is something that you can do in a pharmacy in Britain today. Going back to Parkinson's disease and trying to understand it, how do we go from knowing that there's a change in your DNA to actually developing new drugs and understanding what causes Parkinson's? So what we do in my lab is we look at brain cells, and this is a cartoon of a brain cell. If you zoom in on a brain cell into the cell body, there are lots of things going on. These are mitochondria, which hopefully you're familiar with. These are the energy-producing organelles in the cell that produce the energy which allow a cell to function. This is the nucleus, coloured in blue. And in addition to all of those organelles, there's a group of organelles called autophagosomes, which I've just indicated with these sort of blobs here. These are membrane-bound sacs, which basically go around the cell collecting up rubbish. We can look at those in quite a lot of detail. This is a microscope image of a human cell that we grow in the laboratory. This is the nucleus coloured in blue, and the green and red spots are autophagosomes, these sort of little bags that go around the cell collecting up waste. As a sense of scale, this is 5 micrometers. So this is a greatly expanded cell. We developed this image using a technique called confocal microscopy that allows you to look inside cells with quite a lot of resolution. What autophagosomes actually do is a bit like the rubbish trucks that go around collecting your rubbish on bin collection day. And how that actually works is that they form a lipid bilayer. This is a membrane within the cell that has an open end, a bit like a bag. In fact, Claudio Manzoni, who's one of the people in my lab working on this, always thinks that it looks a bit like Pac-Man, and actually, it's quite a good analogy as to what autophagosomes do, because what they do is when they find something that's going wrong, so here's an example of some mitochondria that are broken down, they're not working properly, they might be causing damage to the cell. The autophagosome goes along, gobbles them up, and seals them in. And eventually takes them to a bit of the cell called the lysosome, which is one of the places that can break down and get rid of rubbish within the cell, where a group of specialized proteins called proteases can attack and break down and recycle those broken mitochondria. What does all this have to do with Parkinson's disease? So, what we also know about Parkinson's is that when you look in the brains of people who have died of Parkinson's, their brains are full of sticky lumps of protein called Lewy bodies. This is an image, this is a microscope image from somebody's brain, a human brain, which has been sliced very, very, very thin into a very thin slice, which can then be looked at using a microscope. And so these little black dots here are individual brain cell bodies. These great big lumps that you can see here shouldn't be there. These are lumps of sticky aggregated protein called Lewy bodies, which we think are one of the things that causes brain cells to die. Now, what we think is happening in Parkinson's disease is that these autophagosomes normally go around trying to collect up this aggregated rubbish, this aggregated protein in these sticky lumps that eventually become Lewy bodies. So normally, they go around, collect them up, seal them up, and get rid of them. Now, if the autophagosomes aren't working properly, then what happens is that these sticky lumps build up within the cell. And there's something about the buildup of these sticky proteins that kills those brain cells and eventually causes people to develop the symptoms that we recognize in Parkinson's disease. What we've been doing in my laboratory is looking at LRRK2, this gene that's mutated in lots of people who have Parkinson's. What we think LRRK2 is doing, using a variety of different approaches, including some of the microscopy that I showed you a few slides ago, is that it's working a bit like the local council. So when the bin collection trucks go around collecting up your rubbish, they're told to go on a certain day into certain parts of your town to collect the rubbish by the local council. Now, if the people at the local council aren't doing their job and aren't telling people, the bin collectors, to go around and pick up the rubbish at the right time or at the right place, then the system breaks down. What we think LRRK2 is doing is that it's acting a bit like the local council. It's telling the autophagosomes where to go in the cell and what to collect. So if you have a mutation in LRRK2, that system breaks down and you get an accumulation of these sticky proteins that eventually cause brain cells to die. How does this work in terms of developing new drugs? So what I haven't told you about LRRK2 is that it's a kinase, which is a specialized type of enzyme that controls lots of different pathways in human cells. They're involved in almost all of the cellular functions that you could think of. One of the interesting things about kinases is that they have very specific binding pockets that allow you to design small molecules or drugs that modulate their activity, that can switch the kinase on or switch it off. We've been working in the laboratory with drugs, and these are chemical structures of drugs that have been developed specifically to switch off LRRK2 to prevent it doing the damage that we think results in Parkinson's. This was developed by a researcher at Harvard University over in America, and this was developed by GlaxoSmithKline, the drug company based here in the UK. And the idea of developing these compounds, developing these drugs, is that eventually we can give them to patients and they can switch off LRRK2 in those patients' brains and prevent whatever is going wrong with those mutations causing them to develop Parkinson's. And actually, although these two drugs are being used in labs, they're not being used in clinical trials, the pharmaceutical companies who are involved in this drug development are getting very, very close to carrying out clinical trials, likely to be in the next year or two, actually giving this drug to initially people who don't have Parkinson's to see if it's safe, and then treating people with Parkinson's to see if it can slow down the disease and prevent people getting worse, their symptoms getting worse. Now, as scientists working here at the University of Reading, we lecture on the pharmacy course, but we also try and tell people about our research. These are examples of two of the scientific papers that have been published from my laboratory. Claudia Manzoni is the lady in the laboratory who thinks that autophagosomes look a bit like Pac-Man, and she's really been doing most of the work looking at all of this. So this shows you how research into trying to understand family history and trying to understand genetics actually can feed all the way through into drug development, and eventually, we hope, to developing drugs that will treat and prevent diseases which are causing huge problems in society today. That's all I've got for you, and I think that's probably just about to time, isn't it? So we're all on time there.